The following episode is based on real-life events. Some of the scenes and characters have been modified. Viewer discretion is advised.
Welcome back to UBC Unsolved. This week we're looking at an intriguing case from the biology department of UBC Okanagan. Follow me to see where it is. Sometime between 10 to 11:30 at night, this is where Dr. Plunkett has been murdered. Science 326, the crime scene. Dr. Plunkett was a well-respected professor at UBCO. Unfortunately, with such notoriety came enemies. One night, Dr. Plunkett was working in a lab on third floor sciences. Sometime between 10 to 11:30 at night, someone met Dr. Plunkett in the lab. They had a confrontation that turned deadly. At 11:45 p.m., a poor first-year chemistry student found Dr. Plunkett's body and immediately called 911. Detective Elisa responded to the scene.
I responded to a call at UBCO about a murder or a dead body or something. Whoever did it left like a bunch of physical evidence. They weren't a very good murderer. We found like a bloody Erlenmeyer flask. Anyway, we need to find out if this blood belongs to Dr. Plunkett or the murderer.
Luckily, the forensic techniques for determining the source of the blood wasn't too difficult. Let's check in with our expert witness, Dr. Alil.
To understand the lab techniques used, we need to first elaborate on the basics of DNA testing. For this, we used restriction length fragment polymorphic analysis. Understanding how the DNA of one person or one animal or one fungus differs from that of another is required before you can understand how molecular genetic analysis is carried out. In humans, more than 99% of the DNA does not vary from one person to the next.
So what do these coding regions even do?
These coding regions generally have an important functional value and have been maintained throughout speciation. However, a small percentage of human DNA sequences do vary. These are the polymorphic sequences. The specific polymorphic sequences chosen for forensic typing are known as anonymous sequences. Anonymous sequences are derived from regions of DNA that do not code for any proteins and do not control any known traits. This means that researchers are able to preserve the confidentiality and privacy of people under investigation.
Sorry, can you summarize RFLP analysis for our viewers here?
RFLP analysis takes advantage of the fact that everyone's DNA varies slightly with respect to the location of certain restriction sites, and that difference can be detected using restriction enzymes. In this method, DNA is cut into fragments using REs, and the resulting fragments are separated on a gel. They can then be stained or labeled radioactively to identify the pattern of fragments and compare one pattern to another. Because it requires a large amount of DNA, RFLP analysis is essentially obsolete in forensics.
We know that the victim tried to fight his attacker and found fragments in the murderer's DNA. We swabbed underneath his fingernails for skin cells and sent the samples down to the boys in the lab. We also found skin cells in the Erlenmeyer flask, the suspected murder weapon.
Very interesting. Now let's check in with Dr. Mandel, our second expert witness.
The results of the forensic analysis were determined using variable number tandem repeats, known as VNTR. These sequences are common to almost all people and are repeated multiple times in an individual's genome. Interestingly, these sequences are the same within the population, but the number of times the sequence is repeated in an individual varies. The number of repeats are inherited in a Mendelian fashion and therefore can vary due to differences between your parents' chromosomes. Repeating sequences observed in this investigation are very specific to the individual and allow the individual to remain anonymous.
So how does VNTR analysis or whatever even work?
VNTR analysis uses restriction endonuclease to cut the DNA in certain ways to allow the number of repeats in an individual's genome to be determined. If there is a small sample of DNA to analyze, PCR can be used to amplify this region. There are some pros and cons to all techniques, but for VNTR, an advantage is that even if you do have a small amount of DNA, you can amplify it through PCR. However, a limitation is that it cannot be used if the DNA is old or degraded, as the VNTR regions must be intact and in the right order. Whoa!
So can you use the VNTR analysis to find the murderer?
Well, you can also use short tandem repeat analysis.
Well, what's that?
I'll tell you. STR analysis is useful when the DNA sample is old or degraded, as only a small length of DNA is required. Another advantage of this analysis is that a small amount of DNA can be used, as again, the target region can be amplified by PCR. STR analysis was conducted for the crime scene DNA as well as for all the 4 suspects we retrieved. The STR region analyzed was the TH01 locus, which contains a repeating TCAT sequence, with the number of repeats varying between individuals. The STR analysis for the crime scene DNA was conducted using the dry skin cells found on the Erlenmeyer flask. Back to you.
Well, let's see if the lab techs can synthesize the answer. Are the suspects they compiled going to be guilty, or are they going to be way off base? And when they find the murderer, how will they react?
To conduct the VNTR and STR analysis, PCR was required to amplify the DNA at the crime scene. PCR works by copying a specific sequence of DNA over and over again. To begin PCR, a template strand containing the DNA sequence of interest, primers that anneal to the beginning of the sequence of interest, a heat-resistant DNA polymerase called Taq polymerase, DNTPs, and magnesium chloride are added to the reaction mixture. There are 4 steps in PCR procedure: denaturation, annealing, extension, and repeat.
Extensions, denaturation, sounds like a bunch of science to me. What are you gonna do with all that DNA?
Denaturation is the first step of the PCR procedure and consists of separating double-stranded DNA into single-stranded DNA using heat. The second step, annealing, is when the DNA primers bind to the complementary section of the template DNA and create a primer DNA hybrid. The heat-resistant Taq polymerase then synthesizes a DNA strand complementary to the target sequence, beginning from the primer bound during the annealing phase. The creation of the DNA strand complementary to the target sequence is referred to as the extension step. The final step in PCR is to repeat the first 3 steps over and over to produce many copies of the same target DNA sequence. A heat-resistant Taq polymerase was used so that the enzyme was not degraded during the high heat of the denaturation step.
So once we have all the DNA, we put it all in the electrophoresis, but we need to know the techniques. Let's check in with Dr. Jelly.
Well, first we need to make a gel. Electrophoresis is a common method for mapping the restriction fragments of DNA or RNA. Fragments are inserted into the gel at the cathode end with an electrical current applied through the gel, which attracts DNA to the anode, and because DNA is negatively charged. A buffer solution is poured over the gel to allow the ions in the solution to carry the charge of the current, which moves the DNA along in the gel.
Sorry, but did you even tell us how to make it?
It's made by combining agarose and TAE buffer into a flask and heating it until the agarose has dissolved. SYPR is then added during cooling to allow the visualization of the DNA fragments under UV light.
Okay, continue on.
The gel is poured into the gel tray and a comb is then added into the gel to create the wells where the DNA will later reside in. Once the gel is solidified, the buffer is applied over the gel, and the DNA fragments from the varying suspects along with the crime scene's DNA are loaded into the wells with the current then being applied. Loading dye is added before electrophoresis and allows us to see the bands.
So how does this DNA move?
DNA moves by size. The smaller the DNA, the quicker and further it moves. This allows us to separate the DNA by bands that correspond to size.
So what's a DNA fingerprint?
A DNA fingerprint is the pattern of bands that results. Because the sizes and therefore the bands differ from person to person, we can compare the suspect's patterns to the pattern of DNA found to figure out which suspect left their DNA at the crime scene.
Okay, last question. What's a DNA ladder?
A DNA ladder of a known size is used to compare the different bands of DNA to use as a reference.
So they had enough evidence to take the murder down, but they need to know if he acted alone. To confirm that no students in Bio393 lab helped, cheek swabs were analyzed. According to a study from Rower done in 2013, only very small amounts of DNA can be extracted from the cheek swabs at a time. You will use the polymerase chain reaction, PCR again, to amplify your sample so there's enough DNA to be visualized. So how do you even store this DNA?
Lots of DNA means lots of power.
CODIS is a software program used to create databases of DNA profiles from suspects, criminals, and crime scenes in many countries around the world. The DNA profiles obtained contain anonymous DNA regions that protect the privacy of the individuals being investigated. As the sequences examined do not give any information that would identify any individual health or medical characteristics. The CODIS system examines 13 anonymous STR loci that are located throughout the human genome.
But can you even tell if it's a boy or a girl?
The amelogenin gene is also examined as it is located on the sex chromosomes and gives information about the sex of the individual being investigated. PCR is used to amplify the amelogenin gene, and different X chromosome and Y chromosome products are created, which are used to classify the gender of an individual. There can be some problems with the sex determination analysis in forensic. These problems arise when there are mutations in the amelogenin gene or when there is a discrepancy between the individual's biological gender and their legal gender. Multiple loci are chosen for the CODIS system because the random match probability, RMP, decreases significantly as the amount of genes examined increases.
You guys have used a lot of big words. All I want to know is, do we have enough evidence to catch this guy?
I think we do. Let's check in with the expert witnesses to see what they came up with. All the forensic testing pointed to one direction: Brendan D'Souza, another lab coordinator. D'Souza admitted immediately to the police. He said that Plunkett had stolen a grant that he deserved. D'Souza was taken into custody April 2nd, 2019, and now is awaiting trial. Well, that shows that murder is no match for good laboratory techniques. Next week at UBCO Unsolved, we look into a case of a disgruntled PhD candidate. See you next time, and stay safe.